Salivary gland. Learning objectives. To study the classification of salivary glands with example, composition of salivary gland, structure of serous, mixed and mucous salivary glands, applied histology, identification points. Parotid gland. Only serous acini are present which contains basophilic zymogen granules and are darkly stained, intercalated and striated intralobular ducts are seen. Interlobular duct can be seen, and previously that was intralobular. It contains adipocytes. Submandibular gland, mixed salivary gland. Predominantly serous with a few mucous acini. Serous demilions are present. Intercalated and striated intralobular ducts are seen. Interlobular duct can be seen. Striated ducts are more prominent than those in parotid gland. Sublingual gland. Predominantly mucous acini are present with few serous acini. Intercalated and interlobular duct can be seen. It also contains adipocytes. Salivary gland. There are two main groups of salivary glands, major and minor. Major salivary glands, three paired glands, parotid, submandibular, and sublingual. They are branched tubulo-alveolar ducts. Minor salivary glands are numerous and widely distributed in mucosa of oral cavity. Some of the minor salivary glands are von Ubbs gland in tongue, buccal gland in cheeks, and labial glands in lips. Structural organization. Basically, a salivary gland consists of stroma, parenchyma, and duct system, which carries the secretion into the oral cavity. Stroma. The connective tissue capsule provides septa and divides the glands into lobes and lobules. The blood vessels and nerves traverse through the connective tissue septa. Large ducts of the gland are also present in it. Parenchyma. Parenchyma has two components, the secretory part and conducting part. Secretory part. Salivary glands are compound tubulo-alveolar gland or racemose glands. Their secretory elements, also referred to as end pieces or as the portio terminalis, may be rounded acini, pear-shaped alveoli, tubular, or a mixture of these, tubulo-acinar, tubulo-alveolar. Salivon basic secretory unit, which includes acini, intercalated duct, and excretory duct. The acini are made up of either serous or mucous cells. A salivary gland may have only one type of acini, or there may be mixture of both serous and mucous acini. These are called mixed acini. A secretory unit or gland. A secretory unit or gland with only one type of cells, serous or mucous, is said to be homocrine. If it contains more than one variety of cells, it is called heterocrine. Serous acini. Serous cells are usually arranged in the form of rounded acini. As a result, each cell is roughly pyramidal, having broad base and narrow apex with a single round and basally located nucleus. The nucleus is pushed to basal region due to accumulated secretory granules of apical region. The apical region is eosinophilic due to accumulation of zymogen granules. Cytoplasm is basophilic due to cell organelle, hence exhibit bipolar staining. Mucous cells. Mucous cells are usually arranged in the form of tubular secretory. The cells lining mucous cells tend to be columnar rather than pyramidal, with a flattened nucleus at the basal part of the cells. The cytoplasm is filled completely with a light staining secretory product called mucus. This pushes the nucleus to the base of the cytoplasm. Lumens of this acini are larger than serous. Seromucous cells. Some salivary glands contain both serous and mucous acini in the same secretory component. They are referred to as seromucous cells. In the submandibular glands, mucous acini are often capped by crescent of or moon-shaped cap of serous cells over them called serous demilunes of Guianeuzi. The serous cells of a demilune drain into the lumen of acinus through the fine canalicula, passing through the interval between mucous cells. Myepithelial cells or basket cells. Myepithelial cells are contractile. The contraction helping to squeeze out secretion from secretory or conducting portion of the gland. They are present in relation to acini. Intercalated intralobular and extralobular ducts. They are located between the lamina and acinar or ductal cells. They contain actin and myosin filaments. Conducting part. The secretory element lead into the series of ducts through which their secretion are poured into the oral cavity. In secretion through the salivary glands, we see a large number of closely packed acini with the ducts scattered between them. These elements are supported by the connective tissue that also divides the gland into lobules and forms capsule around them. Blood vessels, lymphatics, and nerves run in the connective tissue that may at places contain some adipose tissue. Duct system. The ducts are highly branched and range from very small intercalated duct to very large terminal duct. Secretion produced in acini pass along a system of ducts, different parts of which have different structure. The smallest ducts are called intercalated ducts, to which secretory acini are attached. These are lined by cuboidal or flattened cells. Many intercalated ducts join to form striated ducts, and they are lined by cuboidal to low columnar cells. They are also so called because the basal part of the cells show vertical striation. Striated ducts join with each other and form intralobular ducts of increasing caliber. Ducts arising from lobules unite to form interlobular ducts. Striated ducts join with each other and form intralobular ducts of increasing caliber. Ducts arising from lobules unite to form interlobular duct, which in turn form intralobular and interlobular ducts. The interlobular are lined by simple columnar epithelium. Terminal part toward oral cavity gets lined by non-keratinized stratified squamous epithelium. Innervation of salivary glands. Secretion by salivary glands is under hormonal as well as neural control. A local hormone, plasmakinin, formed by secretory cells, influences vasodilation. Salivary glands are innervated by autonomic nerves, both parasympathetic, cholinergic, and sympathetic, adrenergic. Parasympathetic nerves travel to secretory elements along ducts, while sympathetic nerves travel along arteries. Synaptic contact between nerve terminals and effector cells form neuroeffector junction. Two types of junction, epilemal and hypolemal, are present. At epilemal junction, the nerve terminal is separated from the secretoeffector cells by basal lamina. A hypolemal junction, the nerve terminal pierces the basal lamina, comes into direct contact with the effector cells. Nerve impulse reaching one effector cell spread to others through intercellular contact. Classically, salivary secretion has been attributed to parasympathetic stimulation. While this is true, it is believed that sympathetic nerve also excites secretion either directly or by vasodilation. Autonomic nerves not only stimulate secretion but also appear to determine its viscosity and other characteristics. Autonomic nerve terminals are also seen on myoepithelial cells and on cells lining the ducts of salivary glands. The latter probably influence the reabsorption of sodium by cells lining the duct. Salivary ducts are sensitive to pain and must therefore have a sensory innervation as well. Parotid salivary gland.